Hey you guys, today we are talking about the process of mitosis. And this is when the cell divides first. Before the cell divides, the DNA has to divide first. And so mitosis is the division of the genetic material during the cell cycle. And so here, let's think about this before we jump into it. You're made of trillions of cells, but you started out as one single cell. So right here, if we have one single cell, we call that a zygote. So that cell divided into two cells. Here's two cells. And then that cell divided into four. And then four divided into eight. And so on and so on. And so our cells divide and divide and divide until you become trillions of cells. So the sperm that came from your father gave you 23 chromosomes. And the egg that your mom gave you, or that made you up, she gave you 23 chromosomes in that egg. So we have 23 pairs of chromosomes, or 46 total chromosomes. Here's a karyotype, and we have a first chromosome. That one's from mom, and here this one's from dad. And here's our two second chromosomes, one from mom, one from dad. And here's our third chromosome. We have two of those, one from mom, one from dad, all the way till we have 23 pairs of chromosomes. And that is where we start in one cell, and then we divide into two cells and into four. So the zygote went through the cell cycles. That one single cell, the sperm fertilized the egg, and that became one cell. And that cell went through the cell cycle. It went into G1, it grew, it made proteins and organelles, and then it replicated its DNA, and then it went through G2, it grew, it made some more proteins and organelles, and it prepared for M phase. And now here we are at the end of G2 phase, and we are going into cell division, or M phase, where we go through PMAT, prophase, metaphase, anaphase, telophase. And that's how, in PMAT, this is where we divide up our genetic material into two chromosomes. And cytokinesis, this is where we divide the rest of the cell. So mitosis is made up of four parts, and we remember this by PMAT, prophase, metaphase, anaphase, telophase. Prophase, metaphase, anaphase, telophase. And that's mitosis. That's the division of the genetic material. Cytokinesis, over here, so here you can see we have one cell, and then we're dividing up the genetic material. We have one big cell with two sets of genetic material, and then right here, this right here would be cytokinesis because the cells are completely separate. So in mitosis, the type of cells that go through mitosis are called somatic cells. Somatic cells are your body cells. And those are basically every single type of cell in your body except sperm and egg. So your sperm and egg are a little bit different. So if you're a male, you have sperm. If you're a female, you have an egg. And those cells are a little bit different, and we're going to talk about that later. So every other cell in your body essentially is a somatic cell, and that's a body cell. And those somatic cells have the 23 pairs of chromosomes, or 46 chromosomes, 23 from mom and 23 from dad. And so mitosis starts out with one somatic cell, or one cell that has 46 chromosomes. And at the end of mitosis, we have two identical copies of that somatic cell. So here we have a cell that has just two chromosomes in it, just to show as an example. We have one cell that has two chromosomes in it, so let's call that 23 pairs. That's our cell. And at the end of it, we have two daughter cells, two identical copies of that mother cell. And that's the process of cell division. So let's go through and talk about the phases of mitosis. What happens at each phase through PMAT, or prophase, metaphase, anaphase, telophase. During prophase, this happens right after G2. So remember at G2, our cell got bigger. It made extra organelles, it made extra proteins because it was getting ready to divide. And so now our cell is getting ready to divide. We have one set, we have our chromosomes here, And let me grab a draw tool really quick. Here we go. And so right here we can see that there's an X. So one copy of the chromosome. This will be our first chromosome. So we have been, during our S phase, we made copies or we replicated our DNA. And so the replicated, let me use a different color to show our replicated DNA, is right here. So this is the exact copy of that black chromosome. Now we have, so this is one chromosome, but there's two copies of it. So here, let me go back to our black pen. So here's another chromosome, and the exact copy is right here. And so let me do this for the other two. Boom, 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 boom. And let me grab the pink. Where are we? Here and here, here and here. So the chromosomes that are black, those are all going to go to one side of the cell. The exact copies, the ones that are pink, that's going to go to the other side of the cell. And so therefore they can be two identical cells, or they can be two cells that have identical chromosomes. So so here in prophase, if we see right here, we can see, I'm going to grab a draw tool. We can see that, and I'll use purple. We can see that our nuclear envelope, that's the cell membrane, or the membrane that is the nucleus, that holds the nucleus together. And that nuclear envelope is beginning to break down. So I'm going to show you right here, the nuclear envelope. We're representing this breaking down by making a dotted line. So the nucleus is starting to break down. And if you remember that during G2, our chromosomes, or our DNA, is in a relaxed form so that it can be accessed and read, and so that proteins can be made. So right here in prophase, our DNA, or our chromatin, is going to coil up into a more organized form that we call chromosomes. So our chromatin is going to coil up into chromosomes because it's more organized and it's getting ready to divide. Let me go to the next page. So PMAT, that was prophase. Let's look at metaphase. During metaphase, our chromosomes are going to line up in the middle of the cell. So we can see that, let me grab my draw tool again, that this makes it very organized because all of the chromosomes are lined up in the middle, and so the ones that are black are going to go to one side, and then the ones that are pink, so remember these are identical copies, are going to go to the other side. And so this is metaphase. You can see that the chromosomes are lining up along the middle, and so that they're getting ready to be divided. And then you can also see that the spindle fibers, these are spindle fibers, they start from the sides of the cell, and they grow out and attach on to the chromosomes, to one side of those sister chromatids. So remember, when you have two identical copies together, those are sister chromatids. And so those spindle fibers are going to attach on to those sister chromatids, or those chromosomes. So let me draw out the spindle fiber. I'll do it in blue. Here we go. So this is our spindle fiber. It's growing and attaching onto the chromosomes. Here we go. It's growing and it's growing and it's growing. And our spindle fibers have formed something we call a mitotic spindle, and they're attached onto our chromosomes. So this is metaphase. And I like to remember metaphase because the chromosomes line up in the middle, middle. So during metaphase, the chromosomes line up in the middle of the cell. All right, let's go to the next one. So during anaphase, PMAT, prophase, metaphase, anaphase, telophase, I like to remember anaphase in the way that anaphase away. So the A is the same A that I would use in away, because the chromosomes are going to split and move away from one another. So the sister chromatids split into separate chromosomes. We can see, we go back to the black one here, we can see that this was one side, this was one side, right here, and the other side was pink. Here we go, here we go, here we go. And so they're migrating away, anaphase away from one another. They're going to opposite poles of the cell. And you can see how the cell is starting to elongate. It's starting to get longer because it's going to have to separate, and it's going to become two separate cells. So it's getting longer and longer, and that's anaphase. So the two things that happen that I want you to remember during anaphase is that the sister chromatids split into chromosomes, and the chromosomes migrate to the opposite poles. All right, let's go to telophase. We are in the last phase of mitosis, telophase. And telophase is easy to remember because it's just the opposite of prophase. So here we can see that the nuclear envelope, or the nucleus, is starting to show up again. It's starting to rebuild because each of these cells need to protect their chromosomes or their DNA in a nucleus. And so it's starting to rebuild. And then we're getting ready to go back into the G2 phase, or to leave mitosis, and for both of these cells to go into start normal cell life. They're going to be independent cells. And so they need their chromosomes to begin to relax and uncoil and go into the chromatin phase. So these chromosomes are going to relax and uncoil and go into the chromatin phase, so that when we get into G2, they can be accessed and read, and proteins can be made, because our genetic material is nothing more than a set of instructions on how to make proteins. So that's telophase. Let's underline that. Telophase is when our nuclear envelope is building and our chromosomes begin to relax. Let's now, so we're done mitosis. We can see here during telophase we have one cell. It hasn't separated yet. We have one cell that has two sets of DNA, or two sets of chromosomes in it, two identical sets. And when we go to cytokinesis, this is when the entire cell splits, right here. So cytokinesis is not part of mitosis. It is part of M phase, but not mitosis. And so during cytokinesis, the entire cell splits. And so this is going to look different in plants and animals, because plant cells have cell walls and animal cells don't. So animal cells have this right here. We have a cleavage furrow right here. So what happens is there's sort of like this little microscopic piece of string that wraps around the cell. And if you were to think of like a balloon, and if you were to take a string and wrap it around the middle of a balloon and pull, it would eventually sort of pinch, and you wouldn't be able to pull it apart, but it would sort of make two balloons. And that's essentially what happens right here. So we call that a cleavage furrow. We call that a cleavage furrow that we see in animal cells. But plant cells have a cell wall, so we can't use a cleavage furrow. And so what they do is they develop a cell plate, because they have those cell walls. And what that is it's sort of like a scaffold, getting ready for to build that cell wall. So when that cell splits, it's going to have that cellulose, or that fiber cell wall. And so we are building that cell plate, which is getting ready to build a cell wall. And that's how plant and animal cells are different in mitosis. So here we are on our last slide. And we're at the very end of M phase. So cytokinesis, at the end of cytokinesis, we have two cells with identical copies of the genome. The genome is a fancy word for saying your entire genetic complement. All of the DNA that is contained in your cell is called your genome. And remember that all of the DNA that you have in one muscle cell is the same DNA that you have in a brain cell, is the same genome that you have in a skin cell. It's all the same genome in every single cell in your body. And so here you have two cells. So if we started out with that zygote, that one single cell, then it just divided, and now we have two cells. Two cells. So we just, if we go back to this very first picture here, we started out as one cell, and now we have two cells right here. And so both cells are then, so right here we get through cytokinesis, and then both of these cells, here's one cell, are going to start and go into the G1 phase and live a normal cell life. It's going to replicate its DNA. It's going to go through G2, and then it's going to go through M phase again and divide, and then we'll have 4 cells, and so on. All right, well, I hope that helps, and I hope you have a great day. Talk to you guys later.